Okay, hi everyone. This is the third session of the computational spatial biology seminar, and today we're joined by Johann from EPFL. He's going to present VirTues, which is a foundation model framework for analyzing complex tissue architectures. So we're going to find out a little bit more about what that means exactly, and I'll hand it over to Johann. Feel free to ask questions in the chat or unmute yourself at any point to kind of spark some discussion. All right. Take it away.
Yeah, thank you very much for having me. And I'm excited to present today VirTues, AI-powered virtual tissues from spatial proteomics. And yeah, let's dive right into it. The general motivation of this project, maybe as many projects around, is ultimately the better treatment of cancer. And with better, the focus is on more personalized. And what this means specifically, I would like to exemplify with a very simple example in the beginning. Here you can see 3 different biopsies, TMA cores, so tumor microarray cores, of different patients. And all of these tumors are taken from lung tissue of lung adenocarcinoma patients, so the same cancer subtype. And yet these 3 tumors significantly vary. In the left one, you see that the tumor, so it's visualized by the tumor cell type mask. So each color corresponds to a cell type. You see in the left one that the tumor is very compartmentalized in clear tumor areas and some fibroblast immune cell areas, but these areas don't really mix. Whereas on the right side, you see much higher infiltration with immune cells and T cells and much more immune active tumor. And what this is supposed to show is that even though these few tumors are the same cancer type, they vary substantially. And this heterogeneity in terms of spatial architecture matters. So it has been shown in the last years that tumor is not only driven by, and tumor development is not only driven by the genetics of the individual tumor cells and interior mechanisms of a tumor cell, but by the complex microenvironment and interaction of tumor cells and structures around it, such as fibroblasts, vessels, or immune cells in the immune microenvironment. And these structures and this heterogeneity matters also extremely for the tumor development and the clinical decision-making. It has been shown that it affects the immunotherapy response with disease relapse over survival. One very recent and what I found very impressive example here was a study by Gia et al., which was just published a couple of weeks ago, which shows that, for instance, the occurrence of natural killer cells in liver cancer directly affects recurrence of the cancer after resection, and that this information can be used to build a spatial gene panel of 5 gene markers, basically, which allows to distinguish between recurrent and non-recurrent groups of patients with 88.2 hazard ratio, which is really, really valuable information for the clinical decision-making. And for these reasons, it matters that we better understand the tissue structure across scales for better treatment long-term, or better diagnosis, better treatment decision, and better treatment, and hopefully identifying better targets for treatments. And this is the general goal we work towards with our VirTues model. And so the first question is, how do we, in the first place, understand the structure of tissue? And for this, spatial proteomics, just recently declared method of the year, I think, by Nature Methods, is a very valuable tool. We have a general workflow is to follow. A patient has its biopsy taken. The tissue is stained with antibodies. These antibodies bind to specific target markers. And subsequently, using different imaging technologies such as IMC, Codex, or IHC, this tissue biopsy is imaged, and the resulting image looks then, for instance, like this. It's a multiplex image where each channel measures the protein distribution of a single marker. So high intensity areas here in dark blue basically mean the target marker, in this case protein, occurs in a higher rate versus low intensity areas. And so we have now new data modality of, here in this case it's IMC, imaging mass cytometry, where we get very detailed subcellular insights into the tissue, its molecular decomposition, composition, and thus its cell composition and interaction and function. But now we need to interpret this data. And this data is really, really difficult to interpret with just the human eye because of its high dimensionality and poses some new challenges. For instance, these single TMA images are already in spatial dimensions of the 1,000 times 1,000 pixels and dozens of markers deep. For instance, the studies we have been looking at, it was usually around 40 markers. So really high dimensional data samples. At the same time, the cohort sizes, due to the high costs of the technologies, are still relatively small. And we have varying number and combination of measured markers. So one cohort, so study A measures 40 markers, and the next study, depending on that specific target objective of what they want to find out, and the next study might measure another 40 markers, but only partially overlapping. You see this, for instance, here on the right side, where we show the marker panels of 4 studies and datasets we considered in our project, namely one lung cancer dataset, melanoma dataset, and 2 breast cancer datasets. And you see basically each of these bars in the polar plot indicates whether a certain, or each angle corresponds to a marker, and each bar corresponds to whether that marker is measured in the dataset or not. And you see that some markers are measured in all datasets, others only in 1 or 2. And so we need to have architectures or methods which can ultimately be applied to these flexible number and combinations measured markers. And this is what we set out to do. Our goal with VirTues was to build a deep learning, self-supervised representation learning framework to learn virtual representations from this multiplex data applicable to multiple datasets at a time, to learn these representations, and then show and evaluate them on different prediction tasks, which can be for clinical diagnostics, so therapy selection, outcome, or just diagnostics like cancer subtyping or grading, or also just as a tool for biological discovery, for instance, to study and annotate cell types or niches of cells with specific structures or tissue structures. And thirdly, our goal is to, or our vision is that these representations can ultimately be used to build a database of virtual tissues, basically, so that when a new patient comes to the clinic, his tissue can be imaged, compared to past patients' images and tissues, and then the clinical decision-making can be supported by the patient's histories whose tissue is similar to the new patient. This is our general goal with VirTues, what we work towards too. And the centerpiece of the work is basically a new deep learning architecture for this multiplex imaging data and the corresponding training pipeline. And which I will now explain in detail. So we start with the highly multiplex images. And the very first step is besides pre-processing, such as clipping and normalization techniques, that we crop them to smaller areas. This is important because the entire TMA is too high dimensional as a data sample for a deep learning algorithm. We need to focus with the current GPU compute resources we have on a smaller subsection. So our unit of operation on which we operate with our model is basically one crop, which you can see here in dark blue. And this is also what we call a cell niche, and it's a couple of dozen cells, basically. And now our architecture then decomposes this crop image into a 3-dimensional grid of image tokens for tokenization so that we can apply subsequently a transformer-based architecture. So for this, the 3-dimensional, so the multi-channel image is decomposed into its layers, into channels, and each channel is patchified individually. So we introduce one patch per channel per spatial grid position. And each of these patches then serves as an image token after flattening. And this will be the input to our transformer. But importantly, the intensity pixel values of patches might look very similar between markers, yet every marker measures a completely different semantic and biological information. So what matters is that these tokens that the transformer model, which by default is equivariant, can distinguish between these tokens and distinguish the different marker origin of the different image tokens. And for this, we use a protein language model, which allows us to embed every protein marker measured into a single vector representation, which we call the marker token. And this we fuse to the image token. So the image tokens of, let's say, histone get fused with the histone marker to identify that this pixel intensity patch corresponded to histone. And this set of tokens is then the input to our transformer model.
Is the fusion just a concatenation?
In this case, it's addition.
Addition. Okay.
Let's say it's first projection to the model dimension and then addition, which by having a high model dimension higher than the patch squared can learn to basically concatenate if needed or put it into different subspaces.
So if I understood well, actually, the protein language model just serves as a positional embedding or not?
It can be seen as this, yes. I think this is the closest analogy. It's supposed to be a positional embedding, a standardized one, and to induce some structural bias into which token belong to which space. But this has to be seen with caution, since structural bias in the protein language model might not necessarily translate to functional bias. So I think this structural bias ability is limited. Might be valuable, but not all powerful.
Sorry, an additional question. The marker tokens, basically the protein language model, this has been trained on some textual data that happened basically outside of VirTues, right?
Yes, this happened outside. We used a pre-trained ESM model here, ESM2, which was basically trained like I think, bird style.
Okay. So
and it's frozen and we don't backpropagate gradients.
Okay, thanks.
Okay, so next step, we have now this sequence of tokens, which are all where we can identify the channel it originates from. This is our base set as an input. To this set, we concatenate basically one additional channel of what we call cell summary tokens. This is one token for each spatial position, like an extra channel, initially initialized with learnable weights. And these tokens are supposed to learn and aggregate basically information into representation within the encoder. So this set of image tokens and initially empty cell summary tokens gets passed into the encoder, encoded, and we get the encoded cell summary tokens. During training, we mask part of the tokens. So masking means in this case simply dropping them from now. They never go into the encoder in the first place. And instead, we have a subsequent decoder, which again is a transformer architecture, which takes as input the visible encoded image tokens and the encoded cell summary tokens and predict the mask tokens. And what's important here to note is that the decoder, while we put all the tokens together into the encoder, so the encoder is able to learn between correlations between different channels and store whatever it learns and sees into the cell summary tokens, the decoder gets only one channel and the cell summary tokens at a time. So let's say we want to decode and reconstruct histone, then the VirTues decoder only gets the encoded, non-masked histone tokens and the cell summary tokens as an input, but it doesn't get any other markers as an input. So all the information needed has to come from what is visible about histone or what has been stored into the cell summary tokens. This is to basically create an information bottleneck, so that the information actually is to be stored in the cell summary tokens.
Just a quick question. Is the decoding is made channel-wise or is combined with all channel markers?
It is done channel-wise. This is what, so it's done channel-wise, and per channel, it has as input only the tokens of that channel and the cell summary tokens.
Okay, thanks.
I also had another question. When you say cell summary tokens, is it just referring to a per patch token? Because if I'm correct, there isn't any like segmentation done during any of this process,
Yes.
right? So there's no guarantee
That's a
like
good-
one patch correlates to a cell, right?
Yeah, that's a good question. It has to do something with how we evaluate it, but you're right in the observation that it's not a one-to-one correspondence. We chose the patch size, so there's approximately one cell.
Okay.
And it's supposed to be the cell level resolution summary, but there's no one-to-one correspondence, unfortunately.
Yeah, makes sense. Thank you.
So another good expression would be to call them patch summary tokens here.
Okay, makes sense.
Then in the encoder, because with this tokenization procedure, which allows the tokenization procedure, was designed so that we can put arbitrary combinations of channels into the model, because the number of channels basically becomes just the sequence length, and transformers can handle arbitrary sequence lengths. We get a large number of tokens per sample, and to handle the computational requirements here, or limitations, we introduced sparse factorized attention mechanisms. So we don't do full attention between all tokens at a time, but either we do marker attentions. It means that at a fixed position, all tokens at the same position are allowed to attend each other across markers, or within each marker, all tokens are allowed to attend each other across space. And this additionally induces some bias that the cell summary tokens are actually corresponding to a specific position, since one cell summary token is not able to attend to let's say, a stone token at a completely different position. And finally, for evaluation, we have different aggregation mechanisms to aggregate the cell summary tokens into niche summary tokens and tissue summary tokens, either self-supervisedly, that means simply by taking a mean operation, or if we have a specific downstream task at hand, through an attention-based attention-weighted aggregation mechanism, which is fine-tuned and trained on the specific task we're aiming for.
You have any example of specific tasks that you're training for to get these attention scores?
I will show some later. For instance, cancer type prediction or cancer grading, or in the type case of niche summary tokens, we some datasets identified interesting structural multicellular structures, which we predicted in a binary manner.
And just why did you introduce another supervision to aggregate and not just taking the attention scores directly computed by the visual transformers such as a softmax of Q transpose K?
I'm guessing those aren't interpretable, right? Within the transformers.
I think what you mean is something like a class token. I think the problem is that we don't have the loss itself doesn't necessarily incentivize a global representation already, compared to maybe if we are using the Dino framework, there would be one class token, which is attending to all other tokens and would be this self-supervisedly trained aggregation. And this we don't have in this framework at hand. So we either rely on the mean operation as an aggregation or on something which is depending on the downstream task.
Okay. Thank you.
Yeah. And so finally, we trained a roughly 50 million parameter instance of VirTues on the 4 datasets we had. I had mentioned before, which comprised a total of 3,470 images, measuring 96 distinct markers. And next I come to, let's look at the results of this training. Quick look at the time. Okay. So the first thing we evaluated was basically just the training of the performance of the model on the training task, given the whole test set. And here I would like to stress that during training, what we did as masking was independent masking. So each channel got masked independently by sampling so for each channel, we sampled a masking ratio between 60 and 100% and chose the positions at which we're going to be masking independently. This leads to the fact that at some or situation where for given target box, for instance, here the red one, in some channels, this position might be masked and others not. So during training, the model can learn both from spatial correlation patterns as well as from marker correlation patterns. And here I'm showing now some examples of the reconstructions. You see in the first column of each example, always the input to the model, so what the model sees. In the second column, what the model predicts. And the third column, the ground truth. And what we can observe here is that the model indeed learns to reconstruct and predict highly or accurate reconstructions of the expression patterns. We see here, for example, in Carf 1, that both the global structure of a tissue and where no expression occurs is correctly predicted, as well as the high intensity, local high intensity peaks, such as this circular structure here. We see the same in histone, the right column, first row, third column. We see that the model is able to histone, which is a nucleus marker. The model is able to reconstruct where the individual nuclei are to be placed. And we also see in the next row, for instance, twist, that this complex of twist expressing cells is correctly reconstructed. So this was the first very positive result, which shows that there are correlation patterns between channels and within channels, which the model can learn to reconstruct at a quite high fidelity. But the next question we were asking was, what did the model actually learn? Did it learn correlations between markers? So is it translating from one marker to another? Or is it getting the global structure of a tissue? Because both could be used in theory in this masking scenario. And to disentangle this, we studied two different scenarios, or we developed two different masking scenarios and studied the performance of these. In the first setting, we chose to mask one marker completely. So here we mask out all patches within this channel, and all other channels become completely visible, and then we reconstruct that marker. And this shows us basically how well it's able to learn correlation patterns between channels. And you see here again the examples. This time the masked input basically is always 0 because we don't see this channel. And we see that the reconstructions are of fairly high quality, actually. So for instance, here CD68, the model is accurately predicting the different high intensity cells expressing this cluster of differentiation. In the first example. In the other examples, for instance, ER, bottom row, first column, we see that the model is accurately predicting where the tissue ER expressing tissue is and predicting the global structure. But finally, this also shows the first limitation of the model. For instance, in the first row, second column, CD248, we see that the model expresses correctly where the expression is. It also expresses some high intensity peaks correctly. For instance, on the top left border, there's a peak, but it misses one intensity peak. You might see it in the middle of a picture. There's basically one cell, which in the original image has a high expression of CD248. And in the prediction, this is not visible anymore, this relatively higher expression. That shows that, the prediction performance of markers depends on the markers and is limited to some degree, given that we have only a snapshot view of 39 other markers. So it learns a lot, but it doesn't learn everything basically. And this was also reflected by looking at the absolute reconstruction losses, which I'm not showing now because it's a bit overwhelming figure, but it shows that the reconstruction quality in these tasks highly dependent on which marker you try to reconstruct. This was our first takeaway, though, that VirTues learns biological relationships across and between markers. Moreover, this task is very informative for representation quality already, because I mentioned that in order to decode, let's say CD68, the decoder only gets the cell summary tokens and the visible tokens of CD68. Since here, none of the tokens of CD68 were visible, the decoder only gets the cell summary tokens. And the ability to reconstruct the expression patterns here shows that the cell summary tokens encoded by VirTues contain a meaningful representation, allowing us to do molecular regression basically of the tissue. So this is basically the first evaluation also of the representation quality. And now, starting with this second alternative mode, namely what spatial correlation patterns VirTues has learned, we also mask certain regions of the tissue completely. So we chose spatial positions at which we masked all channels this time. And here we see again that the model learns spatial patterns of the tissue and is able to reconstruct structures, but with limitations. We see, for instance, in the bottom row very well in HR2 or HLA-DR that the model is able to reconstruct global structures of the tissue and outlines of the tissue. However, the moment the model has to predict finer details, such as the silhouettes of completely masked cells, it starts to struggle and the predictions become more blurry. This shows the limitation, but it's also very reasonable to what we have trained it on, because we trained on an MSE objective, which basically incentivizes the model to learn to predict the expected expression or presence rate of a protein. And due to the non-deterministic structure of tissue, at some point, if you don't have enough visual clues, the expected signal simply becomes a blurry, smooth signal, or is something blurry. And so predicting the best expected signal is doesn't necessarily mean predicting the most realistic looking signal anymore. But nonetheless, we see that VirTues learns spatial correlation patterns. And next I would move to evaluating the representations it learns on more biological or clinical downstream tasks. We did this evaluation across the different scales we're looking at. So we started at the cellular level and then moved to niche level tasks and tissue level tasks. For the cellular level, we take the representations of VirTues, we get the cell summary tokens, and we applied linear probing to predict cell type or cell categories in a multi-class classification manner. Here it's important to note, we spoke about this earlier, we call them cell summary tokens, but they correspond to one patch, and there's no one correspondence from patch to cells. So in order to get the labels for these tokens, our ground truths, we had to make a rule which cell type actually a patch is assigned to. And so what we are predicting here actually is the predominant cell type within each patch. So we know for every pixel which cell type is at a given pixel. And for each patch, we annotate the patch based on the most frequent cell type across its pixels. And this evaluation led to the performance you see here, where we benchmarked against a different self-supervised representation learning model, namely channel agnostic MAE, recently published by, I think, by Kaus et al., and designed for multiplex images, but focus on single cell images. And we observed that our method in general, across cell types and across data sets, our method outperformed the baseline CMAE, leading to higher F1 scores on the cell type prediction tasks, where the ground truth admittedly is noisy due to this lack of one-to-one correspondence. And the ground truth was basically derived by the authors of the different studies using traditional qualitative biology methods, such as segmentation and then gating and clustering operations. And further, we observed in general trend that the performance, so while we outperformed the baseline we set out, we observed a general trend that the performance varies substantially between the different cell types. Here in the CORDS data set, we had a study which was focusing on cancer-associated fibroblasts. And so we had lots of very fine-grained cell type labels for fibroblasts, some very rare. And there was a general trend that the model prediction performance got better on more frequent cell types, such as just tumor cells, and decreased with rare cell types, such as rare CAFs, which improving upon will be, is current and future work.
Did you find that the mispredictions ended up being in similar CAF types, or where the prediction is kind of like completely off?
Yes, I looked at the confusion matrices. I remember that.
Yeah.
I'm not sure if I looked at for this data set of the confusion matrices at this granularity, but in general, the confusion, like the mistakes, seem biologically somewhat plausible. That it used to be cells which might be so often close to each other, like vessels and fibroblasts, or I think, if I remember, or cell types which are close to each other in phenotype or similar to each other.
But just
So yeah,
a question is a limitation of this evaluation is that you can just like that evaluate a cancer type that the model have already seen. Otherwise, how would you train your linear probing if you're not able to encode the cell summary token?
So we do need for this evaluation, we need annotations to do this evaluation. That is the question.
But is the model have been trained on have been ever seen cancer types here, or it's a out of sample?
Only the linear probe model has seen so cancer types. I'm not talking yet about. Right now, I'm focusing on cell types. The model itself, so the encoder-decoder architecture, has not been trained on any annotations. This is fully self-supervised, but the linear probing head, which is a logistic regression algorithm, this one has been trained on the labels given by the studies.
Okay, thank you.
Thank you for the question. Moving on, next we evaluated also the niche level and the tissue level. So for the niche level, we took the cell summary tokens and we applied an attention-based aggregation mechanism, namely UpMil, to predict the niche summary token from the cell summary tokens, and used this for classification, the classification head of whatever task we had at hand. And specifically, in our study, we looked at the Dunbeck et al. Dataset of breast cancer, where the authors identified different multicellular structures associated with a hazard ratio, or increased or decreased hazard ratio. And we tried to predict the presence of these multicellular structures within a niche in a binary manner. So for suppressed expansion structure, we predict whether it's occurring or not occurring within the niche. And we compared against, again, CAMRE and the second baseline ResNet-based architecture recently used in another multiplex imaging application. Again, observing that we generally outperformed these baselines with a higher F1 score. Yeah. And finally, we repeated this for tissue level or patient level tasks by predicting from the cell summary tokens a tissue summary token using attention-based aggregation, and predicting when the clinical labels such as cancer type, grade, or relapse. Where particularly for cancer type, we got particular high performance, again outperforming the deep baselines, which admittedly are already performing quite strongly as well. For instance, here the cancer type task was the prediction of binary classification between adenocarcinoma and squamous cell carcinoma and non-small lung cell cancer, which is a common task in pathology or in digital pathology for benchmarking histology models. Yeah, this was our tissue level evaluation, and this repeated for also other datasets, which you can find in our publication.
Can I ask for that task? Are you doing multitask learning when it comes to this ABMIL, or is it just on a single task at a time?
It's in a single task at a time.
Oh, okay. Did you happen to, so like I know one proponent of attention-based MIL is that the attention scores hopefully are meant to be interpretable across the different cell summary tokens. I was wondering if you happen to look into those to see like which cell or like which patches kind of corresponded with high prediction of cancer grade, et cetera.
We did something very similar. In the next slide, we were inspecting the attention. This great transition. We were inspecting the attention or factorized attention scores, which the model produced after pre-training. And we were looking either at the spatial attention mechanism. So given that we have both spatial attention mechanisms, where the tokens attend spatially, or marker attention, where they attend across markers, we analyzed them separately. For spatial attention, we used the pre-trained encoder. We added a class token, and we fine-tuned the encoder on the cancer type prediction and looked at what the class token was attending to. And here we saw that you can see this in the left image for one sample. Light means low attention scores and dark means high attention scores, and outline cells are cancer types. And we see that on this task of cell cancer subtyping, the model generally attends more to cancer cells within the tumor versus non-cancer cells. And these get higher attention scores. And similar, for marker attention, we looked at the non-fine-tuned model, and we looked at what the different markers attend, or what the cell summary token attends to in terms of which markers, depending on which crop we input. And we see that, for instance, if we input a crop, the first one here, with lots of tumor cells, shown in dark blue, the model shows high attention to tumor-associated markers, such as PAN-CK in MMP11. So basically here, these bar plots show the top 5 attended to markers. We show that here PAN-CK gets most attention paid to, followed by VIM, CD45I, and MMP11. And if we move to an area where we have more fibroblasts, these attention scores shift, and we see more attention to a fibroblast-associated markers such as VIM or SMA or collagen. And finally, if we move to an area with lots of immune and T cells, we see that immune cell markers get high attention paid to. Yes. Next, we tested our model for this envisioned task of information retrieval in a clinical workflow. So how well our presentations help us to build a database and do information retrieval. So given a new patient, we want to get this representation. We embed it, we get the niche summary tokens using a simple mean aggregation of the patch summary or cell summary tokens. And now we want to query the database we've built with past tissues to find similar tissues. And we chose to do this. So to query a database, we need some notion of similarity between tissues. We present the tissue as a set of niche summary tokens. And to compute the similarity metric, we compute the optimal transport distance, namely the 2-Wasserstein metric, between these sets of niche summary tokens. And this serves as our notion of distance. And we retrieve the top 3 candidates or closest matches. And here you see some examples of these retrieval mechanisms, and more examples which show even better the high variance. More examples you can find on paper, but here you see some examples which show that the model retrieves tissues with similar structure and composition as the query tissue. For instance, in the first row, you see tissues with compartmentalized larger tumor areas and compartmentalized fibroblast areas. The bar plots on the side show the proportion of the different cell types, and you see that the retrieved tissues all show the similar composition in terms of proportions of cell types, and this pattern of compartmentalized tumor cells and compartmentalized fibroblasts. And in the second row, you see a different tissue with a higher immune cell infiltration, and subsequently candidates which are retrieved with also higher immune cell infiltration. And we also analyze the matches in terms of clinical labels and observe statistical correlation that the retrieved patients had similar labels to the query patient. Yeah, so this was our evaluation of that we can use this representations to build a database and do some information retrieval on it. And hopefully at some point this will become useful for the clinic to find similar patients. which might be a nice interpretable intermediate step before full automation of a clinical routine, which might be further away due to the risks. Because in this retrieval task, there's still a clinician in the loop, which can then study the actual difference in tissues and make a decision whether the past patient's histories might actually be helpful, not to inform the clinical decision making. And finally, we were wondering and asking whether our model is applicable also to datasets outside of the training, because given that every study has a different marker panel, in order to build a method which can be trained once and applied also in the future cases, which might be slightly different in terms of which where the tissue was taken from, which exact cancer phenotype the patient has. It's important that the model is robust to such distribution shifts and different marker panels. And so we evaluated the zero-shot capabilities. The idea here, or what we did here, was we trained on 3 datasets, I mean the lung dataset and the breast cancer datasets, and evaluated on the metastatic melanoma cancer dataset and on a diabetic pancreas dataset. And in the first step, we focused on evaluating on these new datasets, but with seen markers. So let's say here we have the melanoma dataset, which contained 40 channels, or roughly 40 markers. A subset of these markers was also measured in some of these training datasets, and another subset was not. And we restricted the dataset to the subset which was seen during training in the first step. And looking at the reconstructions, we see that the model here is able to, again, reconstruct the tissue at a high fidelity. So it accurately reconstructs both single cell structures and single cell expression, high intensity expressions, as well as global structures. And this across all 3 different masking modes. And the difference between zero-shot and trained in terms of quality of reconstructions is relatively small. Looking at the reconstruction loss, we see that this confirms that this qualitative expression, that the reconstruction difference is small between trained and zero-shot evaluation. Sorry. With one exception, namely the marker masking mode. In the marker masking, there the quality of reconstruction deteriorates in the zero-shot mode, which makes a lot of sense, given that we are now on a different tissue site or different organ, namely skin, melanoma, versus lung and breast. And even though the marker was seen, it was always seen in a different setting, in a different combination of other markers. And so the global marker correlations the model might have learned and needs to do marker reconstruction might not perfectly hold in this new dataset anymore, leading to this drop in performance.
It's a little surprising to me that both the marker and niche masking end up having a higher MS, or actually, so lower is better in this case. Is that right for this graph?
Yeah, lower is better.
Oh, okay, I see. I guess it is interesting to me that the niche masking doesn't see a zero-shot drop. Do you have any intuition why that is?
I think my general intuition is that the model has learned in the niche setting, it cannot really base its reconstructions on correlation between markers in the first place. So it reduces basically to only a pattern matching algorithm or pattern correlation learner. And so since these patterns might look quite similar in the new channel, it makes sense that it doesn't drop so much. I think what's interesting is I think that usually the reconstructions for marker masking look better than the niche masking, because the niche does get a bit blurry. The marker does not, which I believe lies in the fact that in the marker masking, it does very well on the relative prediction, but it can shift in the absolute value it predicts on the absolute base level.
I see. for the independent masking, for any given tissue, how many, like for in this example, do you happen to know how many markers that you have? Because I'm imagining at the rate that you're masking, the coverage must be pretty good in terms of every patch most likely has at least one marker that was not masked for that given niche.
Yes, much more. I think the every patch is masked with an expected probability of 80%.
Okay.
So I think masking all patches at a given spot across, let's say, 40 markers gets very improbable.
Yeah.
0 to the power of 40. 0.8 to the power of 40. So usually the model has,
something to work on. Yeah.
Some markers to work with, at each position.
Okay, makes sense.
Which I think is important to keep in mind, and will be good to memorize for the next experiment. So here, to recall, here we are working on a new data set. We've seen markers. We finally also evaluated on cell level predictions and saw that the performance was quite similar between zero shot and trained model. Excuse me. Next, we moved on to evaluating on a new data set, again, this melanoma data set, but also including the new markers and focusing on the performance on new markers never seen during training. And here I show the results for this experiment. I show again independent masking and marker masking. And here we see for the first time in marker masking a significant drop. Suddenly MLANA, melanoma masking, is not accurately predicted anymore if it's fully masked, which is, I hope, not surprising given that this marker has never been seen during training. So the model can't really know how it has to correlate with other markers, except for this structural bias of the protein language model, which is limited in terms of functional meaning. But the moment we move from marker masking to independent masking, suddenly the performance increases again significantly. Suddenly we are able to predict MLANA well. And I think this shows, we interpret this as the model is learning not only correlation patterns between markers on the training set, which hold for all images, but it's also able to detect image specifically and on the fly, or sample specifically on the fly, correlation patterns between channels. Because for every patch here of MLANA, where it sees MLANA, it likely also sees some other visible patches in independent masking. It can see the correlation between these patches and use this basically on the fly in context detected correlations to reconstruct the remaining MLANA. So basically it has learned some in context detection capabilities. And this, I think, is also indicative of why the protein language model, despite its limited structural bias, is really helpful because it allows us in the first place to give a semantic distinguishable meaning and identity to each channel, which the model would otherwise not have. So even though it might not know a priori the correlations of MLANA of other channels, it can, thanks to the protein language model, identify MLANA as a new marker and on the fly detect its correlation patterns. Yeah, with this, I think I come to the final observation of my talk. Finally, I would like to acknowledge and thank my collaborators, because this was a group effort with great collaborators. Ishan, my co-first author, Kirill, Matteo, Andreas Vicky, Gabriele Gut, and my supervisor, Charlotte Bunne. Thank you very much for them, and thank you very much for your attention, and I look forward to further questions or discussion.
Yeah, thank you so much. This was a great talk, and there's a ton of analyses, so it was a really clear picture that you presented of the capabilities of the model, so that was great. So we can definitely pick up questions as we go, but maybe just to start from my side, I was curious about in terms of using the model in practice, especially with these like tissue and niche level prediction tasks. I was wondering, like your main benchmark was this CAMAE, which is, if I understand correctly, also operating on a similar like patch level tokenization and then doing mask autoencoder to do prediction. So it sounds like the pre-processing was kind of held consistent there. But I was wondering if there was, if you had an understanding of how good the prediction was compared to perhaps very simple baselines, such as like in the very beginning of your presentation, you showed like 3 different types of tissues, and one indicator was the structure, but also you could just clearly see that within this tissue, there was a clear distinction in the cell type composition, and perhaps that could be, you know, a set of features that you can select on and potentially do pretty well on prediction. So I was wondering if you had a sense of like how much like delta improvement that you get by doing this like vision transformer model on top of the same data.
It's a good question. We focused mainly on this representation of benchmark against other self-supervised representation learning approaches. I think one large, I did not test this. For instance, you mean cell type composition could be used for tissue level tasks, right? For predicting the cancer type, you could look at the cell type composition. Right. Yeah. I have not done this experiment. It would be interesting. What we benchmarked against was, of course, also let's just taking the unprocessed expression of the process, but not encoded expression values. Mm-hmm. Those we generally outperformed. For instance, for cell type segmentation, we could also just take the raw patch intensity values. Although it's actually a strong baseline, but we generally outperformed it. I think what's a very hard baseline and what we are aiming towards to is that for every task, there's often a specialized model doing it. So for instance, for cell type annotations, we're trying to predict, well, there's no natural ground truth of cell types except what other models have predicted before. Either experts have annotated, but no expert annotates 5 million cells. So usually it's what other models have predicted before. And there are specialized workflows which are a bit more complicated to set up, which define basically our ground truth. And this is where we are trying towards to catching up with. Because this is basically what currently the 100% would be meaning, predicting the same as what the segmentation and clustering plus gating plus random forest would have annotated. In terms of other baselines, none come to my mind for which we have tested. That's a great question.
Yeah, that makes sense.
Makes sense to do that.
Maybe there's 2 questions in the chat. Maybe we can answer these and then finish up. So the first one is about the query retrieval system. I also found this quite interesting. Did you test whether the retrieved datasets had the same niches or microarchitecture as query, or did they just have similar global cell type proportions? I think I also recall you showed like the tissue or sample level labels, right, of what kind of cancer they were and the severity.
Yeah. In the paper, we compare the similarity and the retrieval based on the clinical features matches and the cell type proportion distances. We don't test for niches. It would be interesting. Unfortunately, we did this mainly on the CORDS data set, where we don't have niche annotations. So on CORDS at all, which is our largest data set, which was best suited to build a database, basically, a sample database on the test set, where we didn't have niche annotations. But it would be interesting to see. That's a good proposal.
Yeah, definitely. I also kind of imagine it would be cool if you had like experts, try to label what are the most similar tissues in your opinion and see how close you get to those labels. But of course, that is probably an ambitious experiment to try.
Yeah. I think, on a global, like thinking about just the general direction of this research field, I think what's really a bit missing yet, and what we tried to define in the first place, but as computer scientists and not biologists, was standardized benchmarks. What can we test such models on? What do we want to build these models for? And I think this is still, we made some step towards in proposing tasks, but this is still missing compared to, let's say, classical or digital pathology models for histology, where there's a whole suite of benchmarks already on which models can be compared against each other.
And the last question being, so I think like, okay, well, I think you kind of answered this question. I think I asked the same question where the spatial attention that you're showing was not from the ABMIL, but from this like class token based attention. Is that right?
Yeah, it wasn't ABM. We were injecting a class token into the encoder and fine tuning on this prediction using the class token. And I think it was for the spatial attention was the last or 10 or second to last layer attention scores we're showing.
Got it. And then the last question, okay, yeah, the new last question is for new markers, do you have to add a new channel, or do you just take the protein language model and then take the new vector and pass it
in? Generally, the model can, from a technical perspective, the model can be applied off the shelf to a new data set with new markers, as long as you compute the protein language model embedding. Thanks to this transform architecture, which doesn't take comparison to CNNs, a fixed number of channels input.
Yeah.
So this was one key like feature we wanted to work towards to, that the architecture can support this long term.
Got it. Yeah, so I think with that, we're kind of finished on time. Thanks so much again for the presentation. It's really great. And for people watching, make sure to join next week where we're going to talk to Christina about this model called Cortex.